We're going to do one of the favorite topics of all time in medicine. We have 3.5 hours to do all of human and medical genetics. How many people feel that we'll pull that off? 1, 2, 3. Okay, good. See if the clickers work. I don't know. It doesn't look like they have it turned on. Did they turn it off again? You know? Okay, so this is the problem we're going to run into. Well, forget clickers. Doesn't look like they activated it. This is where we get really messy, nature, nurture. And this is where genetics, you're going to find out genetics, sadly, has become a really big part of medicine. It's often in one type of inheritance called multifactorial, which means that we are clueless as to exactly how it is played with, but it's an environmental genetic kind of interplay that we see going on with it. This is what we're going to cover. Hopefully in response, hopefully this helps you a little bit. But we're going to hit definitions. We're going to talk about gene silencing, which is part of the definitions. We're going to get from base pairs to proteins. Anybody remember how you do that? We got 1. Anything in Worcester? We got 1 in Worcester. 2 and 3 in Worcester. We're going to relocation of genetic materials, so aneuploidy, translocation, deletions. We're going to cover that. We're going to talk about Mendelian inheritance-ish. There's a reason I put the ish in, we'll cover. So inheritance of traits, penetrance, and expression. Population genetics, we're not going to spend a lot of time on it, but you do need to understand, why does a gene stay? Why does an autosomal recessive gene that is deleterious in the homozygous state stay within a population? It should go away, right? Something bad should go away. Well, there are reasons that the genetic material won't go away. Nature versus nurture. We have epigenetics and imprinting. And today, hopefully, we'll just get started on pedigrees. And then tomorrow, we'll take the second side of the slide. But you need to understand pedigrees a little bit for you to have any chance on the second part. So if you don't remember your pedigrees, do a little bit of homework tonight. Just a little bit, not a lot. Anybody know? Okay. Yeah, I don't. We'll see if we. Oh, there's a response. We're up now. Okay. Quickly. If you don't know, just hit a number, because we'll go over it. In the end, I'm going to give you the answers, but not right now. If you're really nice to me, I might actually give you these slides with the answers. But you're going to have to be really nice to me. Like really nice. Like act like you're interested. Enthusiastic, ask questions, clarification. Faster, faster. Okay, so we have both campuses. Do you guys remember what the user feedback thing is? Is that where you tell me where to go or something? It's up now. It just came out. It didn't work on the first slide, and I don't know why. but I'm getting answers from both campuses. Okay. I was about to say, if it's gone, we're really in trouble. Yeah. Okay. Everybody who has a clicker, are you done? Okay. We have 80 responses out of more than that, I hope. So, do we have consistency in our definition, in our response? Not yet. Until 2012, www.microsort.net website provided info for only culling out sperm with 2 X chromosomes. In other words, throwing away the double Xers. Prior to artificial insemination, allowing parents the chance to select female offspring, avoid children with Down syndrome, minimize the risk of having a child with an X-linked trait, reduce chances of matriarchal lineage continuity, or increase relative sperm weight. Ten, 9, 8, 7, 6, 5, 4, 3. It's kind of neat to hear yourself talk. I hate it, actually. Well, we found, I don't know, 5 more people? Cool. Using the Hardy-Weinberg equation, if the prevalence of an autosomal recessive trait is 1 in 100, what percent of the population would have only the normal allele? Only the normal allele. Just punch it, but people who know it, go ahead and figure it out. Just everybody else punch. Come on, you can do it. You can punch. You got 1 through 7 you can push. You can even push 8 if you want to. That's a pretty even distribution, don't you think? Let's try it now where you give me the percent of the population that are carriers of the autosomal recessive. Push the same one if you want to. That's fine. I can tell who knows the answer, by the way, when I look it up. Pretty even distribution. What is wrong with only calculates carrier frequency? Does somebody actually look at the notes? That's pretty cool. And one more. What percent of the population would have a normal phenotype? Aren't you intrigued with these questions? Aren't you bored out of your mind? You get tired. In what situation would genetic counseling provide limited benefit? This is a negative question. Limited benefit. It would not help you very much. Documented genetic condition in the family belonging to a high-risk ethnic group, young maternal age regardless of father's age, consanguinity, or repeated spontaneous aborted pregnancies. The answers to these, by the way, will be at the end of tomorrow's lecture. You won't see these until the end of tomorrow. That's not bad, guys. You're doing all right. You're getting the idea. This sweet, affectionate child, which most of these kids really are, there are very few of these kids to be found for adoption because they're so sought after by adopting families. Not true for every congenital condition, but for this one, it's very hard to find an adoption. This child came through child most likely came by her looks through. Both parents are carriers, a metabolic disorder, non-disjunction, teratogen exposure, or advanced paternal age. A little bit of random choicing here. This pedigree of cystic fibrosis shows which inheritance: X-linked, autosomal recessive, autosomal dominant, X-linked recessive. Bad luck. or a little bit of fuzziness. You're getting in the right direction, guys. Well, 49% of you. In the previous pedigree, the symbol represents, it's not Prince, the actor, the singer formerly known as Prince. No. The symbol represents individuals with partial trait expression, short necks, hemianopsia, normal phenotype, full penetrance, or 50% expression. How many people absolutely know the answer to this?
I assume I know.
Thank you. I don't. But we have a little bit of distance to go then. Okay. Both genetic syndromes shown with it have the same chromosome 15 deletion. Why so different outcomes? Father carried the fetus to term. First trimester prenatal infection. Deletion obtained from mother versus father. And Venetian moon phase shift. Which quite honestly can be a lot of what medicine is about. What the Venetian moon is up to. Excellent. Did we really know that, or we just figured out that's the only possible answer that made sense? Okay. Clinical genetics, the way it was taught to me, the way it was taught to most all of you initially, was that we really targeted very rare conditions, single gene diseases. So you take a single gene, single allele, single part of the chromosome, and you blast it. And that's what genetics was. Only about 10% of pediatric admissions and childhood mortality, however, is related to single gene traits. Polygenic, multifactorial, complex disorders are really what we're looking at 90% of the time. Virtually every medical condition has some genetic component problem. And hypertension, heart disease, asthma, diabetes, mental illness, and these are very strongly known. Depression, bipolar, schizophrenia have genetic basis to them. Learning developmental disabilities, ADD, dyslexia, autism. Old fathers is the current blame for a lot of autism. I think the guys just aren't smart enough to get married early enough, or they can't find somebody to put up with their tendencies. We'll talk about Asperger's later on. Cancer. Cancer is genetic. It's a somatic deletion, somatic duplication. It's a cell gone bad, and it's almost always genetically based. And if it gets into a sperm line, if it gets into a germ line rather than a somatic cell line, ooh. So these are the ems and ohms. A genome. We have about 3 million base pairs in the human DNA per haploid genome, per one side of it. There are about 23,000, 25,000 protein encoding genes. That's it. That's what we are made of, is those 23 to 25,000. Each of them is a functional unit that regulates transcription. Transcription is from what to what? DNA to RNA. Okay? Then some stuff goes on, a little bit of screwing around goes on, then you get to messenger RNA. And then you go from messenger RNA to protein. And what is that called? Translation. Thank you very much. And it's that functional protein that exerts its effect. Okay? A transcriptome is sort of the whole total set of transcripts that the body has. Okay, these definitions are so we're on the same basis. These are not testable in my book. They're boring. You know I don't ask questions like that. But just so that we're on the same page, I hit you with much worse. Okay, so transcriptomes are expressions of genes modified by external influences. Okay? A proteinome is a protein variation in function expressed by a genome, a cell, a tissue, an organism. And then proteomics Proteomics is that study of it. A metabolome is a complete set of small molecule metabolites. In other words, all the things that scurry around your body doing weird things in protein form. And then you have the study of that. And then we talked about microbiomes before, right? When did we talk about them? No? I had to look too. I couldn't remember when I brought this up. No? Close? Endocarditis. Remember all the bugs that could come and get you, that lived with you? This is from that lecture, by the way. You can get it from anywhere. Now, these are bacterial cells. We're not talking about viruses and fungals. About 10 times more bacterial cells than you have. About 150 times more microbial DNA than your own DNA. We're sort of a trash bin of a whole bunch of organisms. And all those organisms and all those cells and all those proteins are part of what impacts us, what makes up our pattern. Now, prokaryotic cells are smaller. They're about 10 pounds of bacteria in your gut. But this is human DNA, that dot. This is the bacterial DNA around you. That dot gets a lot smaller when you add in fungal and viral. Okay? So we're surrounded by a bunch of DNA. Our DNA is surrounded by a bunch of DNA. About 300 million base pairs. Only about 10% of the DNA is expressed. Again, about 23 to 25,000 genes. So what is the other 90%? Well, they're repetitive, non-repetitive DNA regions that don't encode. They kind of hang around. Okay? A lot of them do serve as structural, a role of packaging. So you have your cytosine, your guanine, your thymine, and adenine. These are your base pairs, right? Everybody remembers these. Just if you want some memory cues. The first part of the alphabet and the last part of the alphabet, they're the two that hang together. And the middle of the alphabet hit together. So cytosine and guanine match up, and thymine and adenine match up. They don't cross-match, right? Remember that? Not at all, right? Then you go down to the double-stranded. These are components. Then you get into these nucleosomes or histones. They're bunched up. And a lot of the structural proteins probably have to do with the way they bunch. Then you can get into the solenoids and all that. And finally, you get to the long Q and the short P part of the chromosomes, arms, right? And the centromere is the middle where they hang out together when they duplicate. All right? Everybody remembers that? Kind of. Wee bit. Everybody remember there was a short and a long arm? Good, we're ahead of it. Now, if you go and you throw a methyl group, anybody remember what a methyl group is? CH3. If we throw that on to cytosine, we actually shut the gene expression down in the adjacent chromosomes, base pairs. So there's a whole bunch of different ways we can alter. We have this genetic material, but not all of it's going to be expressed. Like in females, those types, they're not going to express both X chromosomes, right? If they did, what would happen? They'd be different than they are. That wasn't a judgment comment, but they'd be different because you can only really express one of your chromosomes. So one of the X's has to be silenced, and that's called imprinting. And that's done through methylation, that's done through silencing or changes that occur to the chromosome. There are other ways you can do it other than just these little methylation tricks. Well, cancer cells can be deregulated by changing their methylation. There are also covalent post-translational modifications that become permanent for some gene expression. Histones, the coupled areas of the chromosomes, bunched areas, can be reversibly modified by whether they're acetylated, phosphorylated, or methylated. It's just different ways that we can shut off some genes and then turn them back on by taking deacetylating. Okay? All of this stuff is so you understand there are mechanisms being done, not for memorization, please. Okay? So to give you a little more imaging, this is cytosine. We methylate it. We make this stuff hard to get to. You can't transcribe this stuff or transcript this stuff. We throw a little bit of while we acetylate or phosphorylate or methylate, we open up an area of the chromosome, so now it can be read. Okay? By unmethylating DNA in this case, it allows the transcription to occur. Okay? So there are all sorts of ways you regulate how a chromosome is going to be expressed. Steroids, growth factors, hormones, calcium, cytokines, UV light. All of these can alter gene expression. There are a whole bunch of enhancers, silencers that can occur. But we get out here, and basically whatever is allowed the transcription to occur, the DNA is transcripted into hRNA. There's intronic sequences are cut out. It turns into mRNA, messenger RNA. And this is finally translated into a protein. And then there's post-translational processing that occurs, and the protein becomes functional or not functional if there are issues with the way it was made. And this is what has made you up. This is what influences everything about you as far as how you're made, how you're built, what you look like, how your hormones work, how they rage, how angry you are having to listen to a genetics lecture. It has to do with these little globules floating around, creating the interaction. And it comes from environmental effects. It comes from your internal genetic code. Base pairs, remember, are turned into codons. Codons are sequences of 3, triplets of base pairs. The triplets turn into amino acids. The amino acids turn into your proteins. Okay. This right here is really an amino acid, right? Remember the NH2 and the COOH on the other end? That's an amino acid. Okay. So if I have 4 bases, a triplet of bases, that encodes for a particular amino acid, and I have 4 bases I can work with. What are they again? Adenine, guanine, cytosine. Right. If I do that, I can come up with 64 different patterns that I can have triplets of, right? It's 4 base pairs to the 3rd. But I only want about 20 proteins, right? So it is called a degenerative code. More than one codon can encode for any given amino acid, some more than others, okay? It allows a redundancy, it allows a certain amount of slop, it allows for a lot of mistakes, all right? This is the genetic code. How many people remember this thing? Good. UUU gives you phenylalanine. I can throw in a cytosine at the end and get rid of one of the U's. Now I still have phenylalanine. But if I throw in a G instead, I get leucine. Okay? So this is the pattern of how this is read as far as the translation of codons into amino acids. The initiation codon is the AUG. The termination, the stops, are those 3, UAA, UAG, and UGA. Okay? It's just sort of the pattern that we've gotten into, I guess. How many people need to go over mitosis and meiosis again? Please say no. Okay, these things are YouTube, and they're kind of cute. They show the little and everything going on. If you don't remember it, go ahead and watch those. But it's how we end up with genetic material being passed on. How do you relocate genetic material? Well, you either miss chromosomes, you lose a couple, lose one, or you get extras. That's one way to do it. And that's aneuploidy. With maternal age, we find out there are more and more trisomies that occur. In other words, the divisions don't come out, a chromosome gets stuck to another chromosome, and they end up with 3. What's a very common form? 21. What's another good one? 18. What's another good one?
13.
You don't last very long with 18 and 13 as a rule, though. 21, you last pretty well. Do pretty well. What's another one? Triple X, the super female. Woo, Kleinfelder, not so super female, kind of male actually, but a little bit feminized. XXY. So these are the type of things that you see. You can have trisomies, all of them about 25% of pregnancies. Probability of surviving to term, only 5%. A lot of defective fetuses don't make it. And that's how we call out a lot of the problems that we would see. Doesn't work all the time. If I look around the room, I can see a lot of defects that came through. If I look in Worcester, well, we won't talk about that. But no, but I mean, it really is. We're all carrying a lot of defects with us, as we all know, but some more than others. We won't point them out today. You can get up to triploidy. You can get up to tetraploidy. They don't make it, as a rule, where there's a complete set of 3 haploid. I mean, they've got a lot of chromosomal material. But the total number of abnormalities is probably in the 50% range. And this includes spontaneous abortion, stillbirth, and live birth. The probability of surviving is about 5%. What is the rate of major malformations in all births? Anyone want to guess? Anybody want to completely grope? 3 to 5%. You will find some sort of structural defect or a major malformation in births. I go to 100 deliveries, I'm going to see probably 3 to 5 kids who are defective in some sort of structural way. These are the kids who make it. The clinical phenotypes for aneuploidy, these kind of give it trisomy 13, also called Patnow, not very common. But these are 47 XXs or XYs, and they have an extra 13. Edwards is an 18, trisomy is 21, about 1 in 900. Klinefelter, 1 in 600. Are we diagnosing all the Klinefelters? No, we're not. A lot of them really do look pretty normal, phenotypically. Turner syndrome, these are normal intellect folks. They can't have kids, but you see them. You walk around a mall, and you'll see plenty of people that have Turners. They're doing fine. They're a little short, and they do have some structural issues that can be a problem, and they are sterile. But there are a lot of folks with Turner syndrome that we see. XXX, about 1 in 1,200, you really wouldn't know the phenotype difference for a lot of these folks, I don't think. Trisomy 13, they have a lot of central issues in their brain and head development. Their biggest issue is they have holoprosencephaly, where they don't cleft their cerebral hemispheres. So they have one hemisphere, but they also get a lot of midline defects. Cyclops, one eye, a lot of clefting issues. Their average median survival is about 2.5 days. They don't do well. Only about 1 in 20 will survive half a year as a rule. Rocker bottom feet they talk about, they just have, there's really no, well, there's a problem. The flat feet discussion, most kids are flat foot. And they develop an arch over time. These guys actually have an inverted arch where they look like a rocker foot. Okay, a lot of cardiac defects, a lot of neural tube defects. Trisomy 18, they have this very bizarre clenched fist pattern where the second and fifth overlap the third and fourth. I'm sorry, second and fifth overlap the third and fourth, right? So they have this rather bizarre hand grip, and this actually can be diagnostic for 18, a trisomy 18. They also have this nice rocker bottom look. They'll have they'll have this shelf, this isn't really diagnostic. Now, how do you determine low set ears? You go through the inner canthal to the lateral canthal of the eye, come across, and if the ear doesn't make it above it, that's a low set. Very specific? Say no. Louder. Louder. Yeah. Are low set ears specific to a number of congenital problems? No! Thank you. They also talk about rotation of the ear backward and all. There's, it's a constellation of interesting features as a rule. Down syndrome. Anybody seen anybody with Down syndrome? Anybody have a relative with Down syndrome? Anybody have anybody? Very, very common. Wonderful children. The most loving critters you'll ever find. If we all would go and have this neoteny, looking infantile is what neoteny is, we'd probably have a better planet. But they can have profound mental retardation. They can be mildly retarded. I've heard of situations where a girl wanted to have plastic surgery because she was treated dumber than she was and wanted to get rid of the facial features that everybody assumed she was retarded. Stunted linear growth obesity, a diastasis recti is where the rectal muscles are separated so they bulge out the middle. Flat nose, epicanthal folds, very flat facial. At birth, actually, the best way to diagnose them isn't the single palmar crease. It's actually small ears, smaller than 3 centimeters. That's more reliable. Why do they have a single palmar crease most of the time? Anybody know? No, actually they have brachydactyly, meaning they have very shortened hands. And instead of folding in 2 different ways, if you fold your hand, you'll see you can fold it like this and you can fold it like that. And each of these creases folds differently. If your hand is too short, it just flops and you get a single crease. How many in here and in Worcester have a single palmar crease in at least one hand? Okay, you're all secretive. Doesn't mean you have Downs. It just means your hands are a little bit shorter. But it's very common to see in the normal population as well. Well, at least the PA population. Congenital heart disease, 40%. Anybody remember the defect usually associated? If I said endo, would that help? If I said endocardial, would that help? Would I say, like, you rearrange the cushions on your couch, would that help?
Aortic coaptation.
No, endocardial cushion defect is the most common. It's a central defect is what they have. Klinefelter syndromes, other than thin build and disproportionately long in legs, they can be normal. Phenotype can really be normal. Adolescent boys kind of have female type distribution of pubic hair, testicular dysgenesis can be there. This can actually be one of the big causes for infertility. Gynecomastia is fairly common as well. Have they talked about gynecomastia yet? One of the things, just to, there's no confusion, I've heard a number of the pharmacists say gynecomastia is painful breasts, painful nipples. It's much more profound than that. It's quite substantial breast tissue, can be painful, but it can be devastatingly embarrassing to an adolescent boy. This is no minor issue. So if someone presents it as painful nipples or painful breasts, and they kind of discount it, don't accept that. It can be a major defect for an adolescent boy. Okay. Klinefelter, about 15% are mosaics, meaning that they're not pure XXYs, but some of the cell line is XXY. So the disjunction, non-disjunction that occurred didn't occur right at the beginning, it occurred later on in development. But most are normal intelligence. Really, the IQ is dependent on your breeding stock, not on your XXY, XYY, or XXY so much. Subnormal intelligence, however, is possible. Mental retardation is not uncommon. The really interesting thing is, over half of you won't appreciate this, but they found that the higher number of X chromosomes you have in this condition is associated with your mental deficiency. Now, one of our genetics lectures when I was in medical school, when we weren't PC, I'm very PC, as you know, pointed out that anybody with more than one X has a problem. He was male. So 70% of them do have some minor developmental and learning disabilities. They may exhibit behavioral problems and psychological distress, poor self-esteem. Psychiatric issues, a lot of them tend to be blamed for being very aggressive, but anxiety, depression, neuroses, psychoses. They can have this abnormal development of their molar teeth. Now, I think that's testable, don't you think? No. I mean, it's an interesting finding. A lot of them really have the problems with secondary sexual characteristics being decreased because their androgen is decreased. So they can have sparse body hair, high-pitched voice, and female fat distribution, including the escutcheon with that. Gynecomastia by late puberty in 30 to 50% of them, testicular dysgenesis, and again, this can be one of the major causes for infertility workups. It can be so profound a hypogenital effect that they have ambiguous genitalia, and this is one of the causes for ambiguity. And we really don't have the time in the coursework that we can talk about ambiguous genitalia. I don't think we can get into that in pediatrics, but that's a pretty devastating issue. You have a child born and you can't tell what their sex is. What's the first question parents are asked?
Are you related?
Boy or girl? Little of this, little of that. Doesn't go well. And it's a horribly distressing situation. And then they can have cardiac issues, varicose veins, sort of joint laxity. They're more prone to pulmonary embolus, deep vein thrombosis. There are variants. I put these in more just to show you it can go on forever. But you have a 48XXYY, you have a 48XXX. They become increasingly issues of mental retardation. I mean, count up the X's, guys, with it. The XXXXY has a classic triad of mild to moderate mental retardation. They'll get radioulnar synostosis, fusion, and they are very hypergonadotrophic. Hypogonadism, in other words, they have very unresponsive testes, as a rule. There is the XYY, the super male. These are twin brothers. Guess which one has the XYY? Good answer. The taller one. Yes. Very tall stature, acne, Now you have social and mental difficulties adding in. That one doesn't work with the X chromosome theory. It's questionable, about a 1 to 1,000 to 2,000. So a lot of people are wandering around with interesting patterns that we have not been picked up probably with this. But it's a non-disjunction at the second meiotic division, delayed emotional maturity. Now, a good number of you feel that's the case with an EY, and learning problems in school. I think most of my teachers in elementary school felt the same way about Y chromosomes. Turner syndrome is an XO, and again, a large percentage, 43%, are mosaics. This is a very common mosaic, and so the effectiveness of the phenotype change may be altered by how much mosaicism you have. I mean, if you're 100%, you may have this phenotype. If 10% of your cells have XO, you're going to have a very different phenotype with that. But lymphedema of the feet, come as sausage-like toes when they're born, low hairline, shield chest, very broad chest, narrow hip development. This looks a bit more like a male from the back. The bathing suit would suggest otherwise. Lymphedema in this web neck thing is a very common feature. If you look at these kids in utero, they have a lot of nuchal edema, and this is part of where the web neck comes from. But a lot of the ultrasounds can pick up this lymphedema pretty easily with it. Short stature, they don't really get an adolescent growth spurt. They do have some problems with their dental crowding, malocclusion, ovarian failure. Now they have an elevated LH and FSH. The pituitary is trying to get them to behave and send out some hormones, but the ovaries are not willing. So it's a central or a peripheral hypogonadism. Pubic hair can be normal, androgen. Nails can be hypoplastic or hyperconvex. The web neck is secondary to the lymphedema. They are prone to a cubitus valgus, which is the carrying angle. Have you guys ever looked at the carrying angle? Mine comes down relatively straight. I guess I'm more male than female. But females kind of go out at the angle here. If they didn't, they couldn't clear their hips. So we do have sort of developmental differences that are affected by our hormone patterns. Knees are the same issue. Very large Q angle on the knee, which sets you up for patellofemoral pain because the patella wants to displace laterally. That Q angle has something to do with birth and babies through a pelvis. It's a wider pelvis as part of that. Shield test, lymphedema, we talked about. Hypertension can be a problem with coarctation of the aorta and renal anomalies. Again, most concepti with this do abort. So we're not seeing all the conceptions, we're seeing a small percentage. And then you have your triple X syndrome. Really no unusual physical features, medical problems, because most of the X's are inactivated. They're methylated or they're changed in order to silence them. There is a variable phenotype, tall, behavioral problems, clumsiness, poor coordination, small hands and feet, ovarian failure, and again, mental retardation. Why? Those extra X's. Okay. Questions about aneuploidy? Okay. Some more definitions. A genome, or all the genes or DNA content, we're kind of constructed with 44 autosomes, 22 haploid, 2 sex chromosomes. We also have mitochondrial DNA. Where would that come from? Mother or father? Yeah, we jettison the old guys, set off the chromosomes, and they spit out what's left of that sperm. So you can actually, it's a small amount of genetic material, but you can inherit mama DNA, mitochondrial DNA. So a genotype is your genetic constitution. A variant is some change in that genome. A locus is a specific location on the chromosome. An allele is the different types of material you can have at that locus. Okay? So different genetic patterns within that locus of the chromosome. Polymorphism is sort of the normal variation of that occur at the locus. We're not all the same. We don't look the same. So we have to have some heterogeneity. And it's the polymorphism of those alleles, of that locus, that allow us to be as different as we are. A mutation is when you can inherit a change, a disruption in the DNA strand. A point mutation is a single base pair mutation. A germline occurs within the germline, so it has to be within the egg or sperm that will pass through the egg or sperm, so it's incorporated in the DNA in every part of the body. A somatic cell mutation is really not inheritable. It's going to occur often after you've started to develop, so you wouldn't be fully affected necessarily, but you're not going to pass it along. And mosaicism is when you have a whole bunch of different cell lines. So in a Turner's, you have some percentage, which is XY, XX, and you have some percentage that's XO. And again, the degree of the mosaicism can affect the phenotype, the outcome. Your phenotype is what you look like. Genotype is what your building blocks are. Phenotype is what you look like. A trait is kind of a phenotypic property. So whether you have blue eyes, we all have sort of eyes, hopefully, except for some of us have one in the middle. But a trait would be a feature of the phenotype. Heterogeneity is the different look. Similar phenotypes that occur from different genetic mechanisms. This one I've never quite understood entirely. You can have the same outcome, but it can be from a number of different genetic patterns. Genetic markers tend to be genes that are associated with a trait, that are close to each other on a chromosome, so you can follow the marker, which isn't a concern, for instance, a phenotypic problem, but it will show a certain trait that's often associated with another finding. So if you can follow the markers, sometimes you can also follow the trait along. It usually has to do with how close they are on the chromosome together. And translocation is when you pass material between chromosomes. Okay? Again, definitions only. So we're going to go to the relocation of genetic material on the other side of this diagram. So we're going to have translocation, deletion, inversion, and duplication from the wild type. This is homologous chromosomes. So these are chromatids. They've duplicated. No crossover. Nothing happened. Very rare, actually, for this to occur. Two of the genes cross over. And so when they're done, they've switched these portions of the chromosome, okay? You get a double crossover, and you've only switched the parts within the crossover before it recrossed over, okay? So you can reassort all sorts of different ways with that. You all are familiar with this, right? Anybody not? Okay. So reciprocal translocations, chromosome 20 here throws a little bit of its long arm, replacing a large part of the long arm of chromosome 4, and what comes out is very balanced genetic material. It's just on the wrong chromosome. So you probably wouldn't see a whole lot of change in the way somebody looked. It's balanced. There's a weird translocation that it can occur, Robertsonian translocation, where it clefts off at the centromere, at this part right here, in little tiny chromosomes that have a short arm that has very little genetic material. And the two long arms join together, and the small arms are jettisoned. So you end up with one chromosome out of two with both short arms just jettisoned, and you're still balanced more because those short arms don't have a whole lot of material in them. Okay? So what it looks like is this is somebody with a Robertsonian translocation. They got this big mama chromosome. The material that was lost from the two short arms didn't really matter that much. So this person's pretty normal phenotype. But now breed them. They're going to do some interesting things. This is a person with a normal complement of chromosomes. They're going to split off and have one of these, so that looks like their gametes are pretty good. Their sex cells are pretty normal. Now the Robertsonian one can have one of the normal, two of the normals, right? They can also have just this one, fused one. They can have the fused one plus one of the normals or plus one of the other normals. They can have only one of the other. They can have a number of varieties. They can have a normal offspring. But they can also have a balanced carrier. They can have trisomy. They can have monosomy. And they can have a different trisomy and a different monosomy. Okay? These are really kind of interesting to trace down. So there are ways that people will have balances and then they'll go out of balance when they have children as well. But this one, it's just worth knowing about it because it's something that will cause a lot of balanced people to show up. They look fine, and they can't have children. Or they're bad luck. They only give genetic material that gets aborted. So a mutation, again, is any change in the primary nucleotide sequence. It can be silent. There's no consequence. It can be missense, where the DNA sequence occurs in the coding region and alters the amino acid. It may result in a non-functional protein. It can be nonsense, where the reading frame alters and it results in an abnormal protein, almost always, usually unstable. You can have a frame shift mutation, where you take out just a couple or one base pair, or you can have in-frame mutation, where you take out a codon, 3 at a time. And we'll see what happens with those. You can have a splice site alteration, in other words, where the messenger RNA is processed, it can fall apart. You can have a regulatory region, and now you're really screwed up where you just don't express the gene very much because it doesn't even read much of a protein there. So this is wild type. A silent mutation, let's say that you take that G and you turn it into tyrosine. This still codes for the same amino acid. The protein looks the same, so it's silent. This one is a missense. You substitute this G for C. You get a different amino acid here. This can alter this protein substantially. Or it could make a minor change in the protein, but it wouldn't be silent like this one would be. A nonsense would be this G turns into a T, and this is a stop codon. The rest of the protein doesn't even get made. All right? And one base deletion with frame shift, you change this T to a C, or you drop it. Let me see what they did here, actually. They dropped it. All the rest of these amino acids are out of sequence. They're nothing. They don't make any sense. The chance of this turning into a functional protein is nil, almost. Could, but probably not. Okay. We'll finish the missense, nonsense. This nonsense we'll finish, and then give you a break for a few minutes. So a missense mutation, let's suppose this was the genetic code. The big dog has one red eye. Now, how can we tell this is a codon pattern? They're all 3-letter words. Okay? Missense would be the sig dog has one red eye. Not too bad, but it's not the same thing. A nonsense would be the big dog has. It just terminates. Okay? So it gives you a little bit of a sense of how it would affect the transcription. A frame shift would be the big dog has one red eye. We're going to take out an uneven 3. The big dog daha sa her he. Okay? Versus an in frame, we knock out 3. We just knock out a single effect. The dog has one red eye. No big. Does that help a little bit to understand the differences between the frame shifts and the in frame and out of frame and whatever? Okay? 5-minute break, and then we'll make fun of Gregor. Are you lost? Are you lost?
Drinking until I got to school. Oh no, maybe I didn't like that much. It'll be done. I'm gonna finish it.
No, I hear people say that a lot. They don't start drinking until
I do.
He has a really big forehead. You gotta
She's there. She'll take it.
forehead is really big. You kind of grew into it.
Shiver. I got really cold just now.
No, but you know what I mean? If I drink, then I get like tremendous fog. I don't know. Let me do that. Take it. Like
years.
yeah. And you know I'm gonna get bored and I'll be like, oh.
I know. You're gonna walk this like this. And I said like, you know, it's like,
Yeah. I need that. We're leaving.
I can't deal with how it's sunlight in my bedroom again.
Yeah.
God, I thought aliens would
That's like yours too.
I was like, I legitimately thought like someone like lit it up.
You're gonna like open the door and there's all this like wind. It's like a UFO.
Okay. So we know. So we're on the ground floor.
That was the it started from somebody that was involved in the church.
Oh.
That's like
That's not what they said.
I don't think they did anything to him.
They mentioned.
Not exciting though. Double cupping is tacky, don't you know that? Oh god.
What if he's walking in the water at the beach? Pull them up.
Okay, you know what? Lose this.
They are. I imagine Kyle running away.
Frolicking.
Okay. Slowly you will start to see that we actually get into something clinical, I hope. At least what I remember of this. But we're getting rid of the junk, I hope. You may think there's a lot more junk to it, but we'll see. Mendel paid attention to peas, and he was excited about yellow, green, round, and wrinkly. And he did his little Punnett square so that, a dominant trait would give you more purple peas. If you had a heterozygote for the dominant trait, you'd also get purple. And in this recessive pattern, if you had two little bees, you didn't get the pigment, okay? Nice and neat, kind of elegant. He looked at the pollen in the pistil. There's some interesting questions that have arisen, however. Mendel's pea results are considered too good by some extent geneticists. Possible reasons for this statistical improbability: Mendel used a subset of his experiments, presumably the better ones. Geneticists are known to have personality defects. Statistical analysis changed in the interim. Mendel was colorblind, and the results were divine. We're working on your test-taking abilities on this one. Exactly. He faked it. They think he lied. So I don't know. It's a little bit interesting. But the theory is that he did not submit all his results to a panel review, is what happened. Mendelian inheritance. It's a single gene inheritance referred by Mendelian inheritance as transmission pattern he observed on his peas. There are 4 types. This is way too easy. There's autosomal. The gene responsible for the phenotype is located on one of the 22 pairs of autosomes. There's X-linked. It's on one of the X chromosomes, not the Y so much. There's dominant, meaning the conditions are manifest in not only the homozygote for the dominant trait, but the heterozygotes. You only needed 1 copy of the allele to show it. Recessives, you have to have 2 copies of the allele for the recessive trait to show. There are other terms. Penetrance. It's the percentage of individuals having a particular genotype that express the expected phenotype. This is unfortunate to use express in the middle of penetrance because the other one is expression. So don't confuse that. The penetrance is percentage. If you want to try to remember it that way. Penetrance, percentage. PP. If a gene mutation for a disease always shows up with the phenotype, it's fully penetrant. It's always penetrated. If you get the gene, you got it. Okay? Incomplete penetrance are cases of individuals with the abnormal genotype who do not have the disease. There is age-dependent penetrance. In other words, the age that it's expressed can be later. So a delay in the age of onset of the expression of the genetic disease. Huntington's is one of these. So you usually won't see any symptoms until you're at least 30 years old. Okay? This, I'm not going to play it for you right now, but Charles Sabine was a correspondent who debated for a very long time whether to be tested. His brother, his father, or mother. Father died. His brother eventually got the testing because he got symptomatic, and he was sitting on a time bomb, wondering what to do. He finally got tested. He already had his daughter, I believe. Finally got tested, and he's positive. So he's waiting for he knows how he's going to die. Has a bit of an idea when. This is sort of one of those great moments. Who else had Huntington's disease? A very famous performer. Yeah, Arlo Guthrie has waited around for a long time to decide whether he was going to die from this. Eventually they have the genetic testing so they can tell, but it used to be you had to wait and see what was going to happen to you. And all. I think he's been cleared. He's so old now. What does it matter? So your father is just diagnosed with Huntington's disease. What would you do? Get tested absolutely right away. No question. I would think about it for a time. More research is needed before I decide. No, I would let nature take its course. Only if some worrisome symptom occurs. Only if I plan on having a child. Now, Sabine did find out his child is not affected. Okay? Imagine waiting for that test to come back. Interesting. It's sort of, some is the responsibility of the child, other ones just want to know. And I don't know what the right answer is. There is not a right answer. For me, even. Yeah. Luckily, we don't have Huntington's in the family, so that makes it easier. Before you could actually be tested for a lot of these conditions, the age-penetrant diseases, this is the probability that an individual carrying a gene will have developed symptoms by a given age, 40, 50, 60, 70, right here. This is the risk that a healthy child of affected parent carries the disease gene at a given age. So the longer the parent doesn't show symptoms, the less likely you are of having it. This is what Arlo was up against. And this is how Huntington's was decided when I first started practicing before the Human Genome Project that allowed a lot of these things to be picked out. You just waited. Parents healthy. Oh, good. Oh, crap. Now you had to wait and see whether you got the good one or the bad one. As long as the parents stayed healthy, you assumed you were good. So, penetrance versus expression. Penetrance, percentage of individuals having a particular genotype that express the unfortunate, which is an unfortunate term, the expected phenotype. Expression is the degree, how expressive you are. The degree to which the phenotypic characteristic is exhibited. Variable expression refers to the phenomena whereby individuals may experience different signs and symptoms. So you don't always show up with the same gene looking the same. And that's probably a result of environmental factors, other genes interacting, that whole mess of genetics you're mixed up with. So let's say this is the wild type allele, the blue. It's usually the highest frequency in a population. This is the disease right here. The gene function is messed up or it's altered. This is a polymorphism. This is the genes associated with normal gene function, but they're not exactly the same. And these are the ones we haven't a clue what we're dealing with. So there's a whole bunch of different alleles, patterns, and whether they impact, whether they don't impact. Now, we're all assuming when we're talking about the genetics is we know if somebody has the problem, right? Expression tells us that we're going to miss a lot of them. You know, look back on the Klinefelters. Look back on the number of aneuploidy issues, which really can be normal phenotype, but it may not be a problem until they try to reproduce. Okay, so there can be a lot of times we haven't diagnosed the conditions that are expressed by the genes as a result of expression or penetrance. That being said, what are the chances of somebody with an autosomal dominant? It means everybody that gets it has it, right? But they have incomplete penetrance and variable expression. Are you going to see it in every generation? No. But it's an autosomal dominant. You got to, right? No. So reading pedigrees can be very, very creative sometimes. Mutation rates, oogenesis versus spermatogenesis. Now, women pretty much generate their oocytes in only about 20 cell divisions. Okay? There are a lot of mutational rates per cell division. Spermatogenesis, you have about 30 divisions by the time of puberty, and you just throw in another 20 cell divisions every year thereafter. Tell me who screws up the most as they get older. Sperm. Non-disjunction, however, is the woman's fault. Okay? More likely. But bad genetics, bad sequences is often the man's. Okay? It's about 1 in 10 sperm. So women, if you're smart, check their sperm. Check about 10%. You got a better chance. Okay? Any question on that? We're going to do a little bit of population genetics. Please do not panic. How many people remember the Hardy-Weinberg equation? Nobody. Oh, you do? Thank you. Thank you. Population genetics. I'm doing this not because I want to torture you, because I think you need to understand why does a gene stay in the population when it's not good? When it's a bad gene, why isn't it thrown out? Okay? There are reasons, actually. Not always good ones. Let's assume there are only 2 alleles. How often is that the case at a locus? Well, you can have the wild type, you can have the heterogeneity stuff. I mean, often there are more than 2 alleles, but we're going to stay with 2. Otherwise, the math becomes difficult. The frequency of genotypes. Genotype is what you look like or what you're composed of. Okay, your genetics is 2p plus 2pq plus pq squared, p squared, q squared equals 1. This would be the dominant, say the AA. This would be the little a. This would be the carrier. Okay? P squared represents a frequency of AA. 2pq represents a frequency of a little a. And q squared is little a. So if we know the frequency of any allele of a 2-allele system, we can figure this all out. We can figure out how many carriers there are. We can figure out how many people are going to be affected. We can figure out how many people only would carry the non-mutant allele or the non-problem allele. Again, this goes back to the same. It's the same Punnett square issue that we saw with good old Gregor. Okay? This time we're not cheating, though. Most mutations have a negative effect on fitness. Fitness is your relative vigor. So you have a lethal mutation, you die, right? Not good. Bad. But mutations that do not affect fitness can just hang out. If they don't change your fitness, if they don't have a selection coefficient against you, they can just hang out because they're not really being selected against. Some mutations are advantageous. They help you. You lead to the point where you reproduce more. Okay? What are going to happen to those alleles? They're going to have a population pressure, a selection pressure that will increase them, right? This W is what we always talk about fitness. It's 1 minus your selection coefficient. And your selection coefficient is how much you lose by having that gene. So if you have a nastier selection coefficient against you, this is looking at the frequency of the dominant allele. Where there's not a whole lot of selection, your fitness is pretty good. It takes a much longer time for that dominant, the dominant allele to disappear, okay, to become more prevalent. Selection against a dominant allele, in other words, it's not good. If you have less selection, you're going to see it go down much faster. Much stronger selection, you're going to see that allele disappear faster, okay? Don't try to figure this out right now. Look at it, because it makes a little more sense when you start to really dissect it. But the more you're selected against, the faster allele will disappear. Dominant is easy. It disappears faster the more disadvantageous it is. Recessive is going, it's gone. Recessives are messier because you don't always see them. They hide within the population. The dominant hides them. The dominant keeps them out of view. So they disappear, but they disappear in a much slower number of generations. If you look at this is going out 1,000 generations, for instance. These just keep on going because there's always the recessives that are hidden by the dominants that can hang out there and stay there. Not quite gone. They just don't disappear entirely. In the previous slide, stated that lethal dominant trait will be eliminated in 1 generation. Okay? What situation would make that rather a fib? A lie. Geneticists are known to have personality disorders. We know about Gregor. Pronouncing geneticists as lethal by itself, even for elocutionists. The trait would die out before 1 generation since non-viable traits in embryos are routinely aborted. The trait may not be expressed until after reproductive age, and women no longer breed with lethally domineering males. You can answer the one you like if you want. Women like lethally domineering males? That scares me. If you can procreate before your bad trait shows up, Huntington's, then you can make more of them, okay? But ones that would produce, for instance, before you can procreate, they're going to go away in a generation as a rule. So, an autosomal recessive condition affects 1 in 10,000 newborns. What is the expected frequency of carriers? Anybody? I don't know. Let's do it. We know that the case 1 in 10,000, so Q squared, is equal to 10 to the minus 4, 1 in 10,000, right? Therefore, Q is equal to 1 in 100. Everybody get that? Yes? No? Anybody not get that? Please be honest. 1 in 100 genes at the A locus are little a. 99 in 100 are the big boy, the big A. Okay? Make sense? The carrier frequency then is 2pq, which is 2 times 99 over 100 times 1 over 100, or about 1 in 50. Got it? This is what genetic counseling is doing all the time. If a parent of a child affected by the above condition remarries, what is the risk of producing an affected child in the new marriage? To produce an affected child, both parents must be carriers. Right? Everybody agree? Autosomal recessive. Therefore, the risk is 1 in 4 of that the two carriers would make a baby. Thus, the overall risk is about 1 in 200. We know that one person's a carrier. In the population, we have about 1 in 50 carriers. And the chance that both of those pass on the recessive trait is 1 in 4. Right? Everybody get it? That is assuming that no family history of the same disease exists in the new spouse's family, that they're just a normal carrier risk. Okay? So it's about a 1 in 200. This is a question that's going to be asked all the time. It's asked every day, thousands of times, by parents wondering whether they should make a baby. Okay? X-linked red-green color blindness affects 1 in 12 British males. What proportion of females will be carriers? And what proportion will be affected females? Well, it's an X-linked. It's not a Q squared, right? It's a Q. So Q is 1 in 12. They don't have another X to go looking at, okay? So P is 11 in 12. 2PQ is 2 times P, which is 11 and 12 over 12, times Q, which is 1 over 12. You multiply that out, it's about 15 out of 100 will be carriers, right? And for female to have it, they've got to have a Q squared, right? Yes? That's about 0.007. This one gets a little messier because, again, males only are dealing with 1 X as opposed to females have 2 X's. That's why females are going to have much less color blindness in the same population with the same carrier frequency. All right? X-linked traits are going to come through much more in the males because we can't cover up that X defect thingy. Okay. Hybrid vigor. How many people have heard of hybrid vigor? How many people have never heard of hybrid vigor? You guys didn't get a master's in anthropology, did you? I don't know. Overdominance is what it is. When the heterozygote has a higher fitness, when they're more fit than either the homozygote either way. So the heterozygote has a better chance of living. Okay? They're going to make more babies. Right? So AA has that much fitness. Little a has this much fitness. Little a has that much fitness. Name a condition. You can look in your notes a couple slides down. Sickle cell. Sick as hell anemia. That's what they called it in Virginia. Healthy people. Now, if there was no malaria, what would what would the fitness here be? No malaria. This would actually be lower fitness than this because these people at low oxygen tension can have problems. They fly in an airplane, they're at high altitude, they get exerted, dehydrated. They do sickle some. So this fitness would be better, but let's throw malaria into the mix. And now the fitness of the heterozygote is better. Okay? So directional selection. If AA has the greater fitness, then A will become fixed. Large A will become fixed. If little, or here they have little a, if the fitness here is better, you're going to fix it to A over here, little a. If overdominant, the heterozygote has the best fitness of all, then you're going to have a stable polymorphism. You're going to head toward an equilibrium of that allele staying in the population because there's an advantage to those people that keeps them around longer to make more babies. Okay? This is just more for people who want to get really weird and look at it, but this actually shows you where it will fix. That the distribution of the alleles will head toward a single number based on its fitness level. Okay? That I'm not as interested in as you need to understand this. Okay? So quickly, sickle cell anemia and thalassemias. Hemoglobin molecule has 4, 2 alpha, 2 betas, right? In thalassemias, the chains are normal, you just don't produce the same numbers, so you end up with an odd number of molecules. The molecule doesn't form right because you don't have enough 2s and two A's and 2 B's to go together. In sickle cell, the hemoglobin molecule is distorted. Alpha thalassemia, you actually have to have 3 or more deletions of the A globulin in order to have moderate severe anemia. Beta thalassemia is a single base substitution that creates it and distorts it. Sickle cell, about 1 in 500 African Americans, is caused by a single missense mutation that substitutes valine for glutamine at position 6 in the beta chain. A single missense. Structural abnormality, it doesn't fold as well. You get these sickle-shaped cells at low oxygen tension. It's not as efficient. They don't travel through the vessels as well. They get a hemoglobin S crystal, a polymerization that causes the sickling to occur. Most common inherited blood disorder in the United States. About 8%, 1 in 12, of African Americans are carriers. 2 million people have the trait. It's a DNA polymorphism. The number of different variations suggests that there are multiple different places it occurred. Where do you think the most likely origin of these mutations would be? Places with malaria. Yeah. And that's what happens. So, I actually meant to take the slide out, but since it's there, we'll talk about it. This is an infarcted bone. The blood supply to this finger was disrupted, and to the bone marrow here, and that bone is infarcted. It lost its oxygen supply, and so it's remodeling. It looks like a moth-eaten lytic process. This is actually something I saw when I did a summer with Smithsonian in the Mediterranean with thalassemia. You see this hair-on spickling of the outer table, and the diploid is of the marrow-producing part of the skull was widened because it needed to produce more hemoglobin, so it expanded the blood-producing portion. And this turned out to be a question on my boards that I got right. It was the first question I was asked. He flipped up this x-ray, said, "What do you think?" And of course, we're supposed to bomb, right? You know, it looks like beta thalassemia. Right then, I think he was going to fail me for cheating. And he goes, "Explain it." "Well, you got expansion of the diploid and the spickling of the outer table." Well, you know, it's kind of what you see with the hemoglobinopathy and all that. And besides, I saw it when I was in the Mediterranean. Oh, okay. He passed me. So there are a bunch of different hemoglobin patterns that affect your malaria tolerance. There's Duffy. There's SCE, thalassemias, both beta and alpha, and also G6PD. It can be affected with this. The red cells of people that have sickle cell do not sickle for any significant degree if they're healthy and they have normal venous oxygen tension. However, when they become infected, this is the trait. This is not the sickler, but the trait with falciparum. The parasite, they deform because the parasite reduces the oxygen tension, and that deformation will mark these cells, and the phagocytes will come and attack them. The spleen will pluck them. So you reduce your malarial load because the red cells that get infected go, "Hey," and the spleen comes along, and the phagocytes come along and beat the snots out of them. Okay. So they clear the parasite quicker. There's selective sickling with a trait, and the burden becomes less as a result. You also get these superoxide radicals and hydrogen peroxide. They tend to retard growth and even kill the parasite as a result when the cells become hypoxic. C has a similar function. They have fewer episodes of malaria, and it's a milder phenotype. If we took away the treatment for sickle cell disease and let those people die off, C would probably become the predominant type of anti-malarial hemoglobin around. With treatment of sickle cell, we have maintained that population non-naturally, I guess would be the term for it. So the way that we intervene can affect how often the genetic stays around. But C is much milder. When you get an SC disease, you get sort of a modified sickle cell disease. Not as severe as SS disease, but it's more than just the heterozygote would be. Thalassemias have the same sort of pattern. They have reactive oxides, oxygen that kills the malaria. It's produced by the hemochromes. So they have their own mechanism of sort of blunting the malarial effect. Again, this is informational to get some relevance into the genetics for you. Okay, we aren't in hematology right now, but this is to get you into the concept that heterozygotes can win. Duffy antigens are resistant to P-Vivax because they can't get into the cell. They need the Duffy antigen to get in. Glucose 6-phosphate dehydrogenase. Basically, the falciparum doesn't grow very well in the erythrocytes. So all of these have some advantage, and that's why we see them within the population. However, with the wrong exposure away from malaria, they become a disease. Depending on, I think it's sulfite. What is it that G6PD runs into? It's a certain medication class. And I'm blocking on it right now. Is it sulfas? Is it sulfas? Yeah, I mean, this group of people really have a hard time with certain classes of antibiotics, for instance. Okay, so let's do a little application. Sickle cell anemia, 2 alleles. Again, there's the Hardy-Weinberg. Sickle cell disease occurs in 1 in 500. African Americans, about 8%, 1 in 12, are carriers. Does that fit? Does it work? Q squared, the 1 in 500. Everybody get that? Yes? Does everybody get the math on that? The sickle cell disease is 1 in 500. Q squared is the frequency of the SS. Right? So let's say the SS is your Q. It's the recessive trait. Q squared equals 1 in 500. Okay? Conceptually, okay? You don't have to get it exactly, but conceptually right now? Then Q is equal to 0.44 square root, right? You don't have to check my math. Then 2PQ, which is what? The carrier frequency is equal to 0.084 if you multiply it out, or about 8%, or about 1 in 12. So this statement agrees with that statement. Okay? Questions? Complaints? Comments? Okay. if you have a Q, which is the frequency of the recessive trait, 1 minus that frequency is P. Right?
Now I understand.
Does that make more sense?
Yeah.
So if you have 20% Q, you have to have 80% P. If you have 15% Q, you have to be 15%. That's, I was going, okay, where'd you come up with 50? 15% Q, it would be 85% P. Okay? Is that making more sense? Then you can plug it into the formulas to find out what the frequency of those traits or those combinations of those traits would be, or carriers, okay? Overdominance involving 2 alleles at a locus will result in elimination of a dominant trait within 50 generations, gender role reversals, an equilibrium in the proportion of alleles seen, increased middle school bullying, elimination of recessive allele after 400 generations. Let's do all of these, actually. Elimination of a dominant trait within 50 generations. Likely? Takes longer. It takes longer, according to that graph, right? Depending on the selection coefficient and health. Gender role reversals, well, whatever. An equilibrium in the proportion of the alleles seen. Overdominance. Yes. Because the heterozygote is healthier, the 2 alleles will come to some agreement as to where they will be, because the person in the middle, the one that has both of them, is healthier, more likely to reproduce than either side. If the dominant allele is clearly healthier than the heterozygote or the homozygote, the other, what's going to happen? The dominant allele is going to come fixed, and it will come out that this allele is the only one you'll ever see eventually, except for some little mishaps over here, okay? Same thing with the other. But it takes recessive alleles a much longer time to go away because they're hidden. The dominant, it's out there being exposed, assuming that it has full penetrance and full expression. The recessive is hidden a little more. Thank you. Very good. But most inherited diseases are not monogenic, neither dominant nor recessive. Nature versus nurture. Scurvy, tuberculosis is environmental the vast majority of time. However, there is some genetic effect. Sickle cell, kind of in here. It's in the middle. It's very genetic, but it's the environment that affects its expression, how often we see it. Down syndrome, phenylketonuria, hemophiliac, these are very genetically based. Environmental doesn't change them that much, okay? Obesity, genetics are the predominant determinants of weight and metabolism. But there's been a dramatic shift. The environment, the exclusion of autos, lack of walkways, escalators, amusements, labor-saving home appliances, fear of crime causing parents to restrict their children from outdoors. It is a genetic condition that's being expressed more because of the environmental changes. ADD. Parents and siblings have a 2 to 8 times more likely to develop ADD than general population. There are actually these genetic markers that are associated with ADHD. However, the way we teach, the way we make people sit still all day, like in PA classrooms, brings out the disorder for people who have that activity problem, that over-focusing issue, that difficulty selectively attending. Okay? And these are the markers. These are odds ratios if you have that genetic marker. Odds ratio if you have 5-HTT. So there's genetics and environment and all of that. Epigenetics is where the heritable changes in a gene expression is caused by mechanisms other than changing the DNA sequence. You have the same DNA sequence, however, it gets methylated differently, histone deacetylation occurs, so the gene is silenced or changed. The gene structure, the sequence is identical, but we've shut down through a silencer, through a gene expression, through regulation. Genomic imprinting is an epigenetic process. Again, methylation can be involved, histone modification, that changes the gene expression, again, without changing the genetic sequence. So imprinting is caused by the epigenetic changes. This is that same slide again. If you methylate, you hide the genetic structure. If you unmethylate here, you transcribe some information. Again, these are the same issues I think we talked about a little bit. Health endpoints, cancer, autoimmune disease, mental disorders, diabetes, methylation. It's whether you transcribe or not. And all these things can have an effect. Drug, environmental chemicals, aging, diet. These are two very neat syndromes. This is Prader-Willi. These are all old photos. These have been around forever. Both of them have to do with chromosome 15 deletions. Sometimes there's not even a deletion there, it turns out. This is a chromosome 15 deletion, but this guy got it because he inherited the 15 chromosome from the father that has the defect. And the imprinting caused a different expression than this one, which was a maternal inheritance. Same exact genetic structure, but the shutting off and turning on was different based on which parent you got it from. Okay? I don't want you, and I don't know how much deeper that all that express, the exact mechanism and why it's maternal. It doesn't really matter for our purposes. But this is Angelman syndrome. This is the happy puppet syndrome. And this is Prader-Willi. Okay. Real quickly, and then you get to quit. How do you represent a genetic trait in a family? Okay, you got this family, you got that family, you got Donald Duck's family. How are you going to express it so that we can all understand what we're doing? And this is something, if you use it within your notes, your note-taking on family history, it's much faster than writing it out if you get used to doing it. Male, female, they married. Well, let's just say they decided to make things of it. Okay? So this is a bond. This is a relationship line. That's a line of descent. This is a sibship line. So we have a male, box head, female, kind of rounded. These are twins because they come off of the sibship line together. This is the proband, often done as a triangle. A lot of people will change that triangle over to whatever the gender is and an arrow so that you actually know the gender. The proband with a triangle, you don't know what that gender that is. So this is what you sort of draw up. So this is dad, mom, older brother, twins, and this is the guy who's affected. This is the guy that we brought them in. Okay? These are descent lines, line of descent, relationship line. Now, these are monozygotic twins, if you draw that bar in. What's wrong with this picture? Yeah, boy-girl monozygotic twins doesn't work, right? So if I saw this, I know the person who drew it is missing something. Okay? You designate generations by large Roman numerals, and you give linear sequencing by regular numbers. So your identification for this person would be I too. Okay? And that's how you can do nomenclature to say who you're talking about as well. These are the symbols that you'll see. Unknown sex, spontaneous abortion. Affected will often be darkened in. Ones that have dots or hash marks. Well, I'm sorry, hash marks are dead. But dots or half moons are carriers sometimes in the recessive patterns. This is mating. This is when you get two matings, you're really mated. You're consanguineous. You're from Arkansas. She went, oh. Yeah. You don't always have to have the same last name. You can have all sorts of nifty things that way. But these you probably should get some familiarity with, just so you understand it. So again, that's the same thing. What if you were double first cousins? I saw this. I was in the geneticist's office, and double first cousins came in, and they wanted to have a kid. What's a double first cousin? Only 4 grandparents. So a simple way would be, here we have 4 grandparents, male, female. They have families, but let's say they had a boy and a girl somewhere along the line. And this family had a boy and a girl somewhere along the line, along with their other kids. Okay? And this girl decided to marry this, make something with this male. And this male decided to make something with this female. Double first cousins. Oh, yeah, this is consanguineous. Not quite Aztec, not quite some other, or Egyptian, but pretty consanguineous here. They each produce a kid. Well, I'm sorry, these are cousins, siblings. These become cousins. So this couple has a girl. This is really hard to draw, by the way. When I was sitting when this was being told, and I'm going, how do I do it without crossing? And I couldn't do it. And these two decide and end up having a boy, and these two want to have kids. Okay, so this is consanguinity. Right? Yes?
Is on the second line, which one is the child? Are they brother and sister?
These are children of these. These are children of these. Does that make sense? These are generational lines of descent. My hope would be that if 1 and 2 had children, they would think twice because they're brother and sister. And if 3 and 4 had children, that would be the same situation. Okay? So this makes them legal. These guys are legal marriage, and these two are legal marriage. This becomes the issue. Okay? So the study question is, what will happen right here? What will come of the proband?
Banjo music.
That's Georgia, I believe. Banjo music comes. Yeah. Double first cousins are planning to have a family. Genetic counseling predicts a risk of less than 10%. Instead of the 25% for an autosomal recessive, right, to occur, because carriers have a risk of 1 in 12 of having an affected offspring. Many autosomal recessive conditions result in non-viable embryos. They offered homage to the gods. Genetic material from 4 grandparents is comparable to that in the general population, and there is no increased risk compared to the general population. Give me your answer, do. Everybody chimed in who wants to? Good job, guys. Basically, we have a way of sorting out bad genetic material. A lot of them don't make it to term. A lot of them have a spontaneous abortion they didn't even know they had because the fetus was non-viable before it even got far enough along to be detectable. Conceivably, right? So we really do filter out a lot of genetic problems before you deliver them, as a rule. This is Cleopatra's lineage. Okay, I gave up after 4 generations. I couldn't figure out what I was marking. This is Cleopatra. This is her aunt. Here's Cleopatra. Now, she hung out with Julius and had Ptolemy 15, otherwise known as Caesarion, I think. Then she hung out with Marcus Antonius, Marcus Anthony. They had the twins, Alexander and then Cleopatra Selene. These were twins, and later on had Ptolemy Philadelphus. Okay, if we go back up here and you try to follow this pedigree, it's really hard to get all these consanguineous lines together. Now, what does this tell you? What is likely to happen in this lineage? I mean, autosomal dominance, forget that. What happens to autosomal recessives when you start seeing this pattern of consanguinity? A lot of autosomal recessives have a chance of surfacing, right? So your final clicker question for the morning. Cleopatra had Caesarion, Ptolemy 15, with Julius Caesar in 46 BC. Then twins Alexander and Cleopatra Selene with Marcus Antonius in 40 BC. And finally Ptolemy Philadelphus in 35, 36, somewhere around there BC. Once Cleopatra Selene came of childbearing age, what are the chances Julius Caesar and her would yield a progeny with a fully penetrant autosomal recessive trait? How many people know the answer to this? Worcester? Oh, we have one. What's the answer? Number 2. Very interesting. Who else has a more correct answer? Yes. 4. We have a 4. How many people would go for 4? All right. What's a better answer? 0. Why would it be 0? That's called a guess. Okay. They're not related? These guys flitted between Egypt and Rome all the time. Went on forever. Well, let's see. Pretty much guessing. The answer is 0. Anybody who remembers their history, he died in 44 BC. We'll finish this up tomorrow. And there was no sperm banking going on.